1/2 of the Nobel Prize in Chemistry for the year 2018 is awarded to Francis H. Arnold for the directed evolution of enzymes. The other half is shared by George P. Smith and Sir Gregory P. for the phage display of peptides and antibodies. Francis Arnold's study was focused on the enzyme, subtilisin, such that it could catalyze the reactions in an organic environment instead of an aqueous one. She achieved this through changing the enzyme's genetic code by inducing mutations. Many variants of subtilisin were produced through introduction of the mutated genes into bacterial cells. Screening was done to select a variant of subtilisin that was most efficient in digesting casein, a milk protein. In secondary screening, the variant that could catalyze best in 35% dimethylformamide, DMF, an organic solvent was selected. In the selected variant, mutations were induced to obtain a variant that would further increase the capability of subtilisin to withstand the organic solvent and catalyze the reactions on casein. After a series of mutations involving 10 unique combinations, the third generation of mutant was obtained. This variant had 256 times powerful catalytic properties than the initial. This resultant enzyme has facilitated faster chemical reactions that require minimal harmful heavy metals. They also produce minimal byproducts, leading to environment-friendly processes and efficient production. George Smith has been long working on bacteriophage, a virus that attacks bacterial cell widely used for medical research like production of antibiotics. His area of interest has been the technique called phage display. This technique involves insertion of an unknown gene into the genome of the bacteriophage. The inserted gene of interest is ligated to a gene that codes for a coat protein. Upon transcription of these genes, the copies of proteins hence synthesized are displayed on its coat. He further proved that antibodies, that are specific in nature, could certainly be used to obtain the desired proteins from the mixture on the coat. This would open the doors to discovery of the unidentified genes of various proteins. Further, other research studies were carried out by various scientists including Gregory Winter to develop new biomolecules using the phage display method. Antibodies are Y-shaped proteins that bind to antigens. Gregory Winter ligated the gene that codes for an antibody of phage protein to the gene that codes for a coat protein. This would allow the phage to display the antibody along with the coat protein. It also helped him to segregate phages that had the phage antibody from the mixture of other antibodies by using phage protein as the antigen. He subsequently demonstrated that the phage display method could be used in the directed evolution of antibodies. Gregory also created a library of phages with billions of antibody variants by inducing mutations. He then selected phages containing those variants that bound specifically to the proteins of his interest. By using disease targets as the antigens, he could find the antibodies that could best fight disease. Before secondary selection, random mutations are introduced in the antibodies of interest. By repeating the cycle of the selection process multiple times, antibodies that bind more and more strongly and with higher specificities could be obtained. Directed evolution has opened up avenues for environment-friendly methods for synthesis of biomolecules. It has also paved way to novel methods of developing pharmaceuticals for the benefit of humankind.